Chapter 10, Muscle Tissue. Today's lecture is going to be a very lengthy lecture. We'll cover myasthenia gravis and why one of the main treatments for this disease is to block something that blocks something that we want to have happen. Why not just activate that thing? This sort of double negative is going to be a pattern we'll see throughout the year. We'll talk about rigor mortis and why muscles tense up after death. I will discuss the term muscle fibers and talk about why our book says fiber while I just say muscle cell. We'll talk about muscular dystrophy, and this will illustrate why it's important to link the cytoskeleton inside the cell to the extracellular matrix outside the cell. We'll do a little comparative anatomy today and discuss turkeys and talk about the molecular differences between light meat and dark meat. We'll talk about fatigue, which is going to be a very complicated subject, but we'll get to discuss this thing called central fatigue, which will be related to chronic fatigue syndrome. We'll talk about antioxidants. These are very popular supplements, but as it turns out, the science suggests they're not all that healthy for us, so we need to understand why. The most important concept for today is exercise and why it makes us healthier. Our current president doesn't believe it. I don't want to get too political, but when somebody so powerful says something so stupid about science, I need to stand up and make some corrections. And you absolutely need to be clear as to why exercise improves human health. We'll talk about warming up and why that can benefit muscles. We'll talk about muscle tone and discuss the before and after pictures that we see in bodybuilding supplements. And lastly, we can discuss multi-joint exercises. The idea here is that working out the legs might improve the strength of the arms. So we need to discuss the science behind why somebody might believe that to be the case and whether it actually works. So to do that, we'll start with a basic overview of the three types of muscle tissue. We'll then start small, looking at some molecules like actin and myosin. We'll then move on and get a little bit bigger and talk about some organelles. And then we'll get a little bit bigger and talk about cells and the difference between fast and slow twitch muscle cells. And then we'll talk about the entire organ, how it can generate ATP and how it can generate force. The three types of muscle tissue are skeletal, cardiac, and smooth muscle. Today's lecture is going to focus most heavily on skeletal muscle tissue. But what we learn today will be relevant when we get to the circulatory system and the digestive tract later this year. And that's because all three muscle types use actin and myosin to generate force. In skeletal and cardiac muscle, actin and myosin is organized into structures called sarcomeres, which give these cells a striped appearance. Of the three types of muscle tissue, only skeletal muscle is voluntary, meaning this is the only one that our brain can consciously control. Skeletal muscle is voluntary. It contracts rapidly and tires easily, meaning I can't keep doing push-ups forever. This is in contrast to my heart, which is going to continue beating 24/7 for the rest of my life. Skeletal muscle is adaptable, which means the more that you work it out, the stronger it gets. It's capable of exerting forces that can be a fraction of an ounce to about 70 pounds. Now, this should bring us to our first question. Obviously, people can lift more than 70 pounds. Sometimes, when we're doing that, we're using more than one muscle. Still, an individual muscle is capable of lifting more than this. So where did this number come from? When we use our muscles, they are applying torque because of where they attach on our bones, which makes them more efficient. If I were to dissect out a muscle and see how much weight it could lift straight upwards, that's where this number comes from. So of course, in the human body, we are capable of lifting more weight than this. You can't just go to the gym tomorrow, pick up the 70-pound dumbbell, lift it, and expect everybody to be amazed that you've lifted the heaviest object that a person could possibly lift. Cardiac muscle is only found in the heart. We'll cover this in greater detail next quarter when we learn about the cardiovascular system. For now, though, you need to pay attention to how actin and myosin work and to what sarcomeres are. Smooth muscle also uses actin and myosin, but it's not arranged into sarcomeres. Therefore, these cells do not appear striped under the microscope. We'll be discussing these in greater detail in the third term when we talk about the digestive tract. Muscles are organs, which means they consist of more than one type of tissue. We'll be focusing heavily on learning about muscle tissue in this chapter, but keep in mind muscles also contain a fair amount of connective tissues, as well as blood vessels and nerve fibers. Welcome to this diagram. It shows up in almost every anatomy book, and I think it's ridiculous. What we're supposed to see here is that if we keep zooming in on a muscle, it still has the same appearance. It has this almost fractal organization to it. This will mean that once we understand the smallest component of a muscle, we'll really understand the entire muscle itself. Let me go through an illustrated diagram instead of this one. Muscles contain individual fascicles. These are distinct units within the organ. Every fascicle, if I were to cut through it, would be composed of muscle cells. These cells are very long and extend the entire length of the muscle itself. If I were to look at a single muscle cell, I would notice it has a striped appearance under the microscope. These stripes are created by the thick and thin filaments made of actin and myosin, which are organized in repeating units called sarcomeres. Once we understand a sarcomere, we'll pretty much understand the entire muscle. These sarcomeres are made of thick and thin filaments. The thin ones are made of a protein called actin, and the thick ones made of a protein called myosin. And that's where we're going to start the lecture, is learning about these two proteins. I'm going to start with a metaphor. Please keep in mind, on an exam, you must tell me about the physiology of muscle tissue, not my metaphors, but this is where we're starting nonetheless. This little blue guy is myosin, and he's going to pull on ropes made of actin, yanking on a weight. He's part of a team of tug-of-war, and I'll actually have a couple of ropes attached to the same weight, and then I'll get a couple more teams facing in the opposite direction, pulling on a different weight. And when all of these guys work together, they're going to pull these weights closer to the midline. This is going to represent one sarcomere. A bunch of myosin groups facing one direction will pull on actin. A bunch of myosin groups facing in the other direction will pull on more actin, and the length of the sarcomere will shorten. Here's a video of a motor protein similar to myosin walking along the cytoskeleton. We have a number of different proteins that can use energy to pull themselves along these filaments. Myosin can use ATP to pull itself along an actin filament. As long as I have a continual supply of ATP, myosin molecules would continue to do this. In skeletal muscle, the myosin molecules are anchored in place. Therefore, when they burn ATP and pull on the actin rope, it's actually the actin that moves, not the myosin. This will be the basis for contraction. Thin filaments are made of actin. This is part of our cytoskeleton that we learned about in a previous chapter. Troponin and tropomyosin are two other molecules that block the active sites on actin. Troponin and tropomyosin are capable of binding to calcium and moving out of the way. and this is what will trigger a muscle contraction. Before we move on, remember that it's important to not have calcium just floating around a cell, especially one like a muscle cell, which is chock full of ATP, and therefore lots of phosphates floating around as well. The thick filaments are made of myosin. Myosin has a head group, which is the part that binds actin, and tails. The thick filaments are organized so that half of the heads are facing in one direction, and on the other side of this group of proteins, the head groups are facing in the opposite direction. Therefore, when the myosin head groups start pulling on actin, we'll see two bundles of actin being pulled closer to the midline. This is our sliding filament theory of muscle contraction. And what that means is that these molecules slide past one another, but that overall this will lead to a reduction in the length of the muscle itself. Now, 100 years ago, somebody looking under the microscope decided to name what he could see based off of whether it was dark or light. And all of those names got abbreviations, which if you speak German probably make total sense. If you know that Z stands for Zwischer, you know exactly where it's located. But for those that don't speak German or don't remember your German very well, that would have been the part in the middle. I'm not going to go through these old-fashioned names. We're talking about the actual molecules, like actin, myosin, and troponin and tropomyosin. To start a contraction, troponin and tropomyosin must first bind calcium. This induces a conformational change in which they get out of the way, exposing active sites on actin. Once that happens, myosin molecules will bind to actin. This will begin our muscle contraction. Next up, myosin head groups ratchet one time. This action requires no energy usage. The head groups were spring-loaded, and once they bind, they pull one time. In order to let go, myosin must bind to ATP, and then we burn the ATP to spring-load the head groups one more time. If actin is still exposed, myosin will bind and ratchet again, and this cycle will continue as long as actin active sites are still exposed and there is plenty of ATP. Therefore, contraction requires energy. We had to burn ATP. It might seem a little backwards that we used that energy to spring-load the head groups, not to pull. Relaxation, on the other hand, is a passive process. In order for a muscle to relax, all you need to do is remove calcium. That will move troponin and tropomyosin back in the way, blocking the active sites, and myosin will no longer be able to pull on actin, and the muscle relaxes. Rigor mortis is a condition that occurs after death, about 12 hours or so. Shortly after death, calcium will start to leak out of the sarcoplasmic reticulum and into the cytoplasm, and when it does so, it will move troponin and tropomyosin out of the way, allowing myosin to bind to actin. However, in the absence of ATP, the myosin will not be able to let go. It will remain attached to actin, and so muscle stiffness sets in. We call this rigor mortis. It's not permanent, and it's not as strong as somebody actively using their muscles to contract. It can be broken with a little bit of massage and manipulation. The sliding filament theory states that when the myosin head groups start pulling on actin filaments, the sarcomere will get shorter. And because sarcomeres are attached to other sarcomeres, the entire muscle will get shorter. So the next part of the metaphor, imagine that all of these guys are standing on ice, holding hands, and they were going to all pull their hands closer to their midline at the same time. The group of these guys would get closer together, and each of them now represents one sarcomere. Of course, this is not quite the way that it happens in a muscle. In a muscle, one end is attached, so that when they all contract, the length of this muscle shortens up in one direction. This is similar to a water rescue, where one person on shore is holding on to a tree, and once you've grabbed your victim, everybody just moves their arms closer to their midline, and this helps bring the person closer to the shore. Therefore, in the sliding filament theory, myosin pulls on actin and filaments slide past one another, but one end of the muscle is going to be fixed. Therefore, the muscle will contract in a single direction. These sarcomeres are what give skeletal and cardiac muscle tissue its stripes under the microscope. They are highly organized sections of actin and myosin with a little bit of overlap. When you see a whole bunch of sarcomeres organized in a muscle cell, the dark bands are where there are thick filaments made of myosin, whereas the light bands are where there are only thin filaments made of the actin. And as you can see here, there's also a region of overlap. It's important to keep in mind that sarcomeres are not flat the way they're depicted in these textbook pictures. They are three-dimensional. And the head groups do not all detach and reattach in unison, but they are offset. The same way that a tug-of-war team might not all be letting go of the rope at the same time to grab a new handhold. Here we have some electron micrographs of sarcomeres. When we look at them from the end view, we can see that they are highly organized into these hexagon-like shapes. There are 6 thin filaments for every one of the bigger thick filaments that we can see. We've covered most of the molecules now. The thick filaments were made of myosin, and those were the motor proteins. The thin filaments were made of actin. These were organized into repeating units called sarcomeres, with the thick filaments in the center, with head groups oriented in opposite directions. The thin filaments were on the edges, and they were attached to more thin filaments of the sarcomeres next door. A bunch of sarcomeres in a row we call a myofibril. It's a long group of proteins. And that is the major thing that you find inside of a skeletal muscle cell. If I bundle a whole bunch of cells together, I could make a fascicle, and if I bundle a few fascicles together, we'll ultimately have the organ itself, a muscle. Just for fun, how many sarcomeres are we talking about? There's about 1 million muscle cells in the gastrocnemius. That's the big fat double-bellied muscle in the back of your calf. Each one of those cells has about 1,000 myofibrils. Those are those long protein strands of sarcomeres. And they're about 15,000 sarcomeres in length. So that's about 15 trillion sarcomeres altogether. So that covers the basics about muscle tissue, as well as most of the molecules that we need to worry about. Actin, myosin, troponin and tropomyosin, the ion calcium, and ATP. Next up, let's talk about muscle cells. These cells are very long. They started off as regular-looking cells that all got together to form what's called a syncytium, a large multinucleated cell, that then proceeded to fill up its cytoplasm with actin and myosin. This left very little room for anything else inside of the cell. All of the nucleuses got pushed to the edges. The book calls these muscle fibers. They are special in that they're not a regular cell. However, the osteoclasts that we discussed in Chapter 6 were also multinucleate, and we still considered those cells. So I'm going to keep using the word muscle cell rather than muscle fiber, and that'll free us up to use the word myofibril without confusion. So the cytoplasm is chock full of myofibrils. Those are the sarcomeres that are attached to each other end on end. Like any cell, muscle cells are surrounded by plasma membrane, and they do contain cytoplasm. The book tries to give these special names, and that's very typical of an anatomy textbook. But in medicine, things get complicated enough without having to give special names to things that we see throughout the human body. So I would recommend just calling the plasma membrane, and the cytoplasm, the cytoplasm. T-tubules are invaginations of the plasma membrane. If you poked your finger into a water balloon without piercing the balloon, you would have created something similar to a T-tubule, an extension of the surface that penetrates deep into the cell. Adjacent to the T-tubules are regions of smooth endoplasmic reticulum known as sarcoplasmic reticulum. These structures are special enough to deserve their own name. This is a place where calcium gets stored within the cell. Here are a couple pictures showing that the T-tubules and the sarcoplasmic reticulum wrapping around the myofibrils. This is going to be important. When the sarcoplasmic reticulum releases calcium, I don't want it just floating around anywhere. It might bump into a phosphate and turn into a crystal. It's right next to the actin and myosin and should very quickly be bound by troponin and tropomyosin. The T-tubules prevent this from happening, a muscle contracting non-uniformly. We're going to receive a signal at a distinct location, but I want all of the myofibrils contracting all at once. I don't want the contraction starting from one side of the cell and then moving to the other side of the cell. It would tear itself apart if that happened. Therefore, I've got extensions of the plasma membrane deep inside of the cell, so that when the surface receives a signal, all of the myofibrils can get that signal more or less at the same time. Here are some pretty pictures showing how organized these things are. To the right, somebody used a green stain that they dumped on top of the cells. This stain could travel down the T-tubules, but it was not able to get across the plasma membrane and into the cell. So we can see how highly organized those T-tubules are. They're very straight and repetitive. Here are some more pretty pictures of the mitochondria, and hopefully you're getting the idea that muscle cells are highly specialized cells. We've got a number of structures, but unlike in BI 112, where it didn't matter where you drew a lysosome or a mitochondria, in a muscle cell it does. Here are a few more pretty pictures where linker proteins were identified that are responsible for ensuring that all of these structures are tightly organized. They fit together kind of like Legos. Our next topic is to cover how muscle cells are instructed to contract. Our nervous system will send an electrical signal down a neuron, which will travel to the end where it connects to a muscle cell, and we call that connection the neuromuscular junction. Acetylcholine is released. This will trigger another electrical signal, which will travel across the plasma membrane and down T-tubules of the muscle cell, which will cause the sarcoplasmic reticulum to release calcium, which will bind to troponin and tropomyosin, which will then allow myosin to bind to actin. So if you didn't get all of that, let's go through it with a little cartoon. The electrical signal travels down this neuron until it reaches the end, and it triggers the release of acetylcholine. This acetylcholine travels just a short distance across the neuromuscular junction, and it binds to acetylcholine receptors. These receptors trigger an electrical signal on the muscle cell, and this electrical signal will travel down the T-tubules, bringing it very close to the sarcoplasmic reticulum. This will trigger the sarcoplasmic reticulum to release calcium. That calcium does not go very far. It is very quickly bound up by troponin and tropomyosin. Those two molecules get out of the way, which allows myosin to bind to actin. The filaments slide past each other, and we get muscle contraction. Now, there's one last step. This is a very important one. We must quickly remove acetylcholine. So there's an enzyme called acetylcholinesterase. I know that's a mouthful, but its function is really important. Otherwise, whenever you contracted a muscle, it would stay contracted. Let's go through these in a little bit more detail. Acetylcholine is a neurotransmitter. It's a chemical released by some neurons. This chemical can bind to proteins on the surface of the muscle cell, which causes them to open up and conduct electricity. As I mentioned on the previous slide, this acetylcholine needs to be broken down very quickly by this enzyme here, acetylcholinesterase. After we've triggered release of calcium from the sarcoplasmic reticulum, it binds to troponin and tropomyosin, which then get out of the way and allow the myosin head groups to bind to the actin filaments. And this starts our contraction cycle. To go back to being in a relaxed state, we need to get rid of this calcium. We can pump some of that back into the sarcoplasmic reticulum, but to get the calcium off of troponin and tropomyosin and allow this muscle cell to relax, we're actually going to trade it for a magnesium ion. Now, this seems like an annoying little bit of chemistry, but if people are deficient in magnesium, they will get muscle cramps. There's a lot of different things that can cause muscle cramps, but this is one of them. The calcium would bind to troponin and tropomyosin, but then have a difficult time letting go. And if the troponin and tropomyosin remain out of the way, the myosin head groups will remain connected to actin. With adequate magnesium, the calcium can be bumped off of troponin and tropomyosin, and it can go back to blocking the active site, preventing myosin from binding, and this muscle relaxes. With myasthenia gravis, the immune system makes antibodies that bind to the acetylcholine receptor and block it from functioning. Because the muscles receive less acetylcholine, they do not contract with as much force. This causes muscle weakness. In turn, the muscles will begin to atrophy for reasons we'll need to understand before the end of this lecture. This condition is treated with a class of drugs called cholinesterase inhibitors. What we want is to make sure that there's as much acetylcholine as possible. But rather than simply injecting acetylcholine into this person, which, even if that would work, would cause all muscles to contract all at once, we instead give them a medication that blocks acetylcholinesterase. So as some acetylcholine gets released, it stays in the synapse for longer, and it tends to build up in the synapse, which should make sure that what few acetylcholine receptors are still functional are functioning at peak activity. So we have blocked something that blocks something. We've blocked the enzyme that breaks down acetylcholine, and this increases the acetylcholine signal. So why this double negative? Well, the acetylcholinesterase inhibitor would be doing nothing at any synapse that was not in use at the time. So it will not increase muscle contraction strength at muscles that were relaxing. If I were to inject somebody with acetylcholine and it traveled to all of their muscles, it would cause all of them to contract at once. In fact, that's what plants have nicotine for. It activates the nicotinic acetylcholine receptor and can cause all the muscles in an insect to contract at once, causing it to fall off the tobacco plant and die. Just for fun, these acetylcholinesterase inhibitors are a medication when used in the proper dosage for the proper patients, but similar chemicals are the organophosphate pesticides, which are pretty nasty chemicals. And these in turn are related to chemical warfare agents like VX gas. In a healthy person, if you were to block acetylcholinesterase, that would cause too much acetylcholine to build up in the synaptic cleft. And every time you contracted a muscle, it would remain contracted for too long. When this is your diaphragm, that means you breathe in, but then you can't breathe back out and you suffocate. So we inhibit inhibitors because the effects can be much more specific than simply activating. The acetylcholinesterase inhibitor would only function at this synapse here that's releasing acetylcholine. This would cause acetylcholine to build up and activate just this target cell. The drug would have no effect at any of those other synapses. Whereas VX gas or the organophosphate pesticides, these would activate all of the acetylcholine receptors everywhere. And this would lead to death. So that's why we use this sort of double negative approach. We're going to see this over and over throughout the year. Myasthenia gravis is not curable. These drugs simply slow down the progression of the disease and can improve muscle strength. With treatments, life expectancy can be average in length. One of the signs of myasthenia gravis is drooping facial muscles. It turns out that a little bit of muscle weakness could easily be compensated for, except in places where muscle tone is very noticeable, such as our facial features. So this might be the first signs that this condition needs to be treated. So that's the neuromuscular junction and how we trigger a muscle contraction and all of the molecules involved in that muscle contraction. And now I'm going to go up and get a drink. You won't know it, but I'll be back in a moment. It would be a good time for you to take a break as well. Let's recap. A muscle is organized into multiple fascicles. Each fascicle is a distinct collection of individual muscle cells. These cells are very long. If we look at them, we would notice that they are full of actin and myosin arranged into myofibrils with repeating structures called sarcomeres. These are an arrangement of thick filaments in the center with thin filaments on the edges. The thick filaments are the ones made of myosin. The thin filaments are the ones made of actin. There's also connective tissue surrounding the muscle. Around the entire muscle, the connective tissue is called epimysium. Around each individual fascicle is perimysium, and between individual cells is endomysium. Within this connective tissue is a significant amount of collagen fibers. These fibers extend to the ends of the muscle, where they become the tendon. So there is no real end to the tendon and the beginning of the muscle. They're both part of the same organ. These collagen fibers in the tendon we call dense regular connective tissue, but then that becomes the periosteum around bones, and some of those collagen fibers even penetrate into the bone as Sharpie's fibers. So this makes for a very strong connection, not over just one little spot, but over the entire diameter and length of the muscle, as well as around the entire surface area of the bone. Collagen doesn't taste very good. Some muscles have more of it than others. For muscles that receive a lot of damage at the surface of the body, they're going to have more collagen fibers than muscles deep inside the body. For this reason, the biceps muscle from a cow is a lot cheaper than its psoas major, a muscle on the insides of the hip bones, where it's nice and protective. Of course, this ignores the flavor of fat when it comes to meat, but that's another topic. Muscular dystrophy is a disease where the actin filaments are not connected to the collagen fibers outside. There is a complex of proteins that help to anchor the cytoskeleton inside the cell to the collagen fibers of the extracellular matrix outside of the cell. It just so happens that one of these tends to get mutated more than others. Mutations in dystrophin can cause the disease muscular dystrophy. The muscle is still able to contract, but it doesn't pull on anything when it does, so it does not generate much force. Because of the reduced amount of force that this muscle generates, this leads to muscle weakness, which will progressively get worse. There are no cures for muscular dystrophy. The best treatments include the use of orthotics and either physical or occupational therapy. There are some treatments on the horizon that may get at the cause of the disease. Rather than go in to try and fix the mutation at the DNA level, these drugs make ribosomes commit errors. One of the most common mutations that causes muscular dystrophy is a premature stop codon halfway in the middle of the mRNA. These drugs would get ribosomes to accidentally mess up and put an amino acid when they should have stopped. And this will get the protein translated in full. It may have a weird mutation in a few spots, but at least it'll be full length. That's much better than half a protein. You may wonder what's going to cause the ribosome to miss the stop codon at the end. Turns out there's actually lots of stop codons at the ends of our mRNAs. So even if it skips over one or two, it'll probably hit the third or the fourth and stop translating the protein. So that's the connective tissue that we find within a muscle. Next up, we can start talking about bigger things. We'll talk about how muscle cells make ATP. This is the energy molecule that muscles use to generate force. When a muscle runs out of ATP, it can quickly regenerate that supply. Used ATP becomes ADP, and muscles have a molecule called creatine phosphate, which can quickly give a phosphate back to the ADP, turning it into a new ATP molecule. Think of this like a battery. It's not energy for free. We had to charge up the creatine phosphate, and we do that when we're resting, but at least I can have energy on demand when I'm working out. Each muscle cell has enough ATP to generate force for about 2 seconds. So there you go. You can go down to the gym, pick out a barbell, hey, maybe that 70-pound one, and lift it for 2 seconds and call it good. You would have burned all of your ATP in the muscle you were working out. Of course, that's not going to be very impressive. You've skipped over the creatine phosphate that's in that muscle cell. With creatine phosphate, you could work out for a further 15 seconds. Still not impressed? Well, muscles contain glycogen, that polymer of glucose. Using this, muscle cells can generate ATP. If they do not have enough oxygen, they'll have to generate ATP anaerobically, and you've got about 2 minutes before you've completely run out of energy. If you do have access to oxygen, muscle cells can undergo aerobic respiration, and that would give you energy for 2400 seconds. Aha, that's only about 40 minutes. Can't most of us work out for longer than 40 minutes if we try? Well, it turns out that this number represents the glycogen that's actually in the muscle. We've got other energy reserves. There's glycogen in the liver. That could be mobilized. But adipose tissue is also another big energy reserve. We could dump some of those fatty acids into the bloodstream and with a trick get them to the muscle cells and burn that for energy. So if that's the goal, you're going to have to burn through the glycogen first before your body would be relying on burning adipose tissue. So we need to cover the basics of respiration. Anaerobic respiration is what cells do when there is not enough oxygen, and it only produces 2 ATP per glucose molecule. Aerobic respiration, on the other hand, generates about 36 ATP for every glucose molecule that is burned. So you may ask yourself, why would I ever bother with anaerobic respiration if it's so inefficient? And the answer to that is, why would you ever drive a car in a race that wasn't, say, a Prius or something with really good gas mileage? If you're in a race, sometimes what you want is explosive energy. It doesn't have to be efficient, but it needs to be explosive. And anaerobic respiration, even though it's not very efficient, it can generate the most ATP all at once to provide maximal force. Aerobic respiration is what cells do when they don't have to undergo peak performance and when they have plenty of oxygen. If you get that luxury, then of course you would want the most energy efficient mode of respiration. So let's review our cellular respiration. We've got mitochondria. At rest, oxygen should be plentiful, and it turns out we prefer burning fatty acids. And so muscle cells, using oxygen, could break down these fatty acids and harness some of the energy in the form of ATP. We could also take glucose from the bloodstream and convert it into glycogen. This is an anabolic reaction, so that would take some energy. If we're undergoing a light workout, we could rely on just our glycogen reserves, as well as oxygen, to provide enough ATP to keep contractions going. It's when we reach peak activity, where we're trying to generate as much force as possible, or when oxygen is not in the bloodstream for whatever reason, that we have to switch over to anaerobic respiration. Here we burn glucose right in the cytoplasm, and it produces just 2 ATP per glucose, but I can do a whole lot of that all at once. It also produces lactic acid as a byproduct, and it turns out it's going to take us energy to get rid of that stuff. But lactic acid very quickly diffuses into the bloodstream and gets carried to the liver, where it can be converted back into pyruvic acid. This slide here shows that there's no switch over from aerobic to anaerobic. As our energy demand increases, we do relatively more and more anaerobic respiration. But this is not a binary process. At rest, when oxygen is plentiful, we're definitely doing aerobic respiration. At peak activity, when you can't provide enough oxygen, we're definitely doing anaerobic respiration. But somewhere in between is probably a mixture of the two. So at rest, all muscle cells prefer burning fatty acids. And during light activity, that's when we might have to switch over to the carbohydrates. And at peak activity, we're definitely breaking down glucose and producing lactic acid as a byproduct. Our muscles feel fatigued when they are not capable of generating the force that we wish them to. I used to believe that this was a simple matter of running out of ATP, but it turns out that muscle fatigue is way more complicated than that. There's a number of factors that will cause a muscle cell to produce less force and less force and less force until we finally say that muscle is fatigued and it can't do what I'm asking it to do. Before we can go into full detail about muscle fatigue, I need to review a little bit about enzyme kinetics. Hopefully in your biochemistry class you learned about ways to speed up enzyme-catalyzed reactions or slow them down. When I teach about enzyme kinetics, I like to use another metaphor, a woodchuck who can chuck wood. That's his enzyme-catalyzed reaction. He will chuck wood from one side of the page to the other, and we could measure the amount of wood that he chucks. Let's say, for instance, that our baseline enzyme rate is about 5 logs per minute. If there were more logs, more substrate, this would speed up the reaction. he wouldn't have to travel as far to get to the next piece of wood and then chuck it. So our rate of reaction would speed up. We would be more efficient. On the other hand, if there were fewer logs, if the substrate was diminished, our woodchuck would have to travel further. And if there's a lot of product building up, then sometimes when I chuck a log, another one just bounces right back. So both of these things can slow down an enzyme-catalyzed reaction. Not enough substrate or too much product. Furthermore, if I dumped a bunch of acid onto our woodchuck here, he wouldn't be very happy, and as he denatures, he would not be able to chuck wood anymore. This too would slow down the rate of our reaction. Now that we have the basics, let's go through some specifics. I did not expect you to memorize these two lists, but for the concepts that I go over here, I do expect you to be able to discuss them on an exam. First off, the reaction that we are concerned about is catalyzed by the enzyme myosin, and the reaction is contraction. This requires the use of ATP, so that's our substrate, and this would be the product here. Things that can lead to muscle fatigue in a high-intensity workout include potassium accumulation. That will make a lot more sense next quarter, so let's skip that one for now. But ADP accumulation is something you should understand. This is the product, or one of the products, of our reaction. So if too much of this builds up in the cytoplasm, even when you have ATP, it could slow down the reaction, leading to fatigue. This next one should be pretty easy. As lactic acid builds up, it denatures our enzyme myosin, which now will not work nearly as efficiently. And this also causes fatigue. For more endurance workouts, fuel depletion can be an issue, meaning we've run out of our ATP supply. Electrolyte loss can also be a problem. Once again, this will make more sense in BI 232, so let's skip that one. But next up, let's talk a little bit about central fatigue. This one isn't understood very well, but what this means is the brain believes that it's tired, and so it sends weaker signals to your muscles. And your muscles behave more weakly, even if they have plenty of ATP. Time for a story. That's me years ago, going for my black belt test. I had to travel down to California for a 2.5-day long event. They had me doing all sorts of stuff. I had to go on runs, do a bunch of sparring, and they had me do push-ups on my knuckles in the parking lot until I couldn't do anymore. Then they asked me to do 100 more. And then at one point they had me stand in what's called a horse stance with my knees at 90-degree angles, with my arms completely outstretched, holding cups of water. Eventually my arms got pretty tired and I spilled my water. But if our arms came down, or if we bent over to relax our legs, one of the black belts would come over and kick us in the stomach. That hurt a lot, so I was very motivated to keep my arms outstretched at all times. But eventually they became fatigued and I couldn't. Even I gave my ki, my little yell to try and move my arms back out again. Eventually I wound up not being able to hold my arms outright at all. It was at that point that one of the instructors came over and said, "Close your eyes. Listen to me walking through the grass, listen to the insects, listen to the creek." I was sure that I was just out of ATP, that it was going to be impossible for me to move my arms outstretched one more time. Believe me, I was very motivated to do so, and I couldn't. Much to my surprise, when I opened my eyes again, my arms were outstretched. I understand a little bit more about human physiology now, and I realize that my brain had undergone central fatigue. It was hot out. I wasn't used to California. I was in this gi made out of thick cotton. I was pretty tired. My brain just stopped sending out signals, even though it knew it would mean me getting kicked in the stomach. And yet a little bit of distraction, and suddenly I could do it again. What I had experienced there was central fatigue, and apparently My instructors were able to trick my brain into forgetting that's what was happening, and my arms were outstretched once again. So, as our muscles are working out, a lot of things decrease the efficiency of this enzyme-catalyzed reaction. Heat is another one that can cause myosin to partially denature. As we get too hot, our muscles behave less efficiently. But central fatigue also involves heat. Our muscles are not only heating up, the entire body heats up. And heat can denature some enzymes. That's fine. I could always make new ones if they totally break down. But the brain is not so happy with that concept. It cannot do without its enzymes, even temporarily. So to prevent the brain from overheating, the brain will undergo central fatigue, meaning the brain thinks that it's tired even when the body isn't tired yet. It will send out weaker signals in an effort to get you to perform less intensely. For this reason, if you go out jogging in the afternoon on a hot day, you're going to get tired a lot sooner. It's not like you're burning ATP faster or any of those other things that could lead to fatigue. The reason that you get tired faster is because the brain starts to overheat and it sends weaker signals to the muscles. And when the muscles are weaker, it feels like you are fatigued. So this is a defense mechanism. It seems highly probable, at least to me, that if the brain can turn on this sort of defense mechanism, that it could do so accidentally and perhaps leave it on long term when it shouldn't. So studying muscle fatigue might tell us quite a lot about conditions like chronic fatigue syndrome, where people feel tired even though they shouldn't. It turns out that this has something to do with serotonin signaling, and we can give drugs to people that shut down central fatigue. To my knowledge, these are not considered performance-enhancing drugs, although they do lead to an increased workout in the heat. The dangerous side effect is that if you allow your brain to overheat, you could do some permanent damage to it. But for people with chronic fatigue syndrome, this may explain why antidepressants can treat their condition. It's not that it's a form of depression, it just uses the same neurotransmitter. Next up, let's cover oxygen debt. If you go out for a really long run, when you get to the finish line and stop running, you are still breathing heavily. We need to ask, why is that? If you're not exercising anymore, why does your body still need extra oxygen? And that's because you've gone into oxygen debt. Your muscles owe that debt to the rest of the body. As they use up oxygen, your lungs try and give the muscles all of the oxygen they need, but if the lungs can't keep up, the muscles can borrow some oxygen from other parts of the body. So as we are working out, not only do the muscles become hypoxic and fatigued, so does the liver and intestines and other organs. For that reason, when I'm done exercising, I still have an increased oxygen demand. I don't need to be generating huge amounts of ATP for exercise, but I do need to replace all of the oxygen that was taken away from my other organs. So I will continue to breathe heavily. If you want to look into this, the technical term for oxygen debt is excess post-oxygen consumption. So that's the phrase you would have to look up. So those are some of the basics of how muscle cells generate ATP. They can either do it with oxygen or without. We also covered some of the basics of fatigue. You should definitely understand why being low on ATP would be a problem, as well as changes to pH or temperature, denaturing enzymes, and we also covered central fatigue. Next, we need to consider muscle strength. This is a very vague term. Muscle strength could be talking about the power of a muscle, how much force it can produce all at once, or how much endurance that muscle has. Both of these factors depend on the types of cells found within the muscle, and what's inside of them. So let's talk a little bit about muscle strength. This will depend on a number of different factors, including the size of the muscle, how fascicles are arranged within the muscle, the size of things called motor units, the summation of those motor units, something called length-tension relationship, and lastly, how fatigued that muscle is. So we need to go through these one at a time, but here's your list of factors to study from. Let's start with fascicle arrangement. Muscles come in different shapes. Two very common shapes are called parallel versus pennate, or fan-shaped. The pennate muscles are a lot stronger. You will see them in places where we need great strength, like in the quadriceps or the biceps brachii. In a parallel muscle, like the tibialis anterior, I've got two skinny tendons, and all of the muscle cells run parallel to one another. So I can only get as many cells as the tendons are wide. So it's not many muscle cells. In a pennate muscle, the tendon actually runs the entire length of the muscle up through the middle. This allows for more attachment points, which means I can fit more muscle cells in this muscle, and the muscle bulges a bit outwards. Furthermore, because of the way that these muscle cells are attached to the tendon, they're at an angle, and that increases their efficiency. So pennate muscles are a lot stronger than the parallel muscles. Next up, muscle cells come in three basic flavors. Different books may count differently. I'm just going to keep it basic. We've got fast twitch fibers, slow twitch fibers, and intermediate fibers. The difference between these different muscle cells is that some are really good at aerobic respiration and others are really good at anaerobic respiration. Please keep in mind I said they are good at. I did not say that's all that they do. That's a very important distinction that you will need to understand by the time we're done with this section. So let's review respiration really quickly. At rest, muscle cells can use oxygen and burn fuel like fatty acids to generate ATP. This is happening primarily in the mitochondria. They can also convert glucose into the polymer glycogen. This requires some ATP to build this macromolecule. Under low amounts of exercise, we can burn some of that glycogen using oxygen to generate more ATP. But under really high activity, if we're pushing this muscle cell to its peak performance, we have to burn those glucose molecules using anaerobic respiration. There's just no time to wait for oxygen to diffuse into a mitochondria, go through that citric acid cycle, and have ATP diffuse back out. I need the ATP in the cytoplasm where the actin and myosin are located. So we generate that ATP using anaerobic respiration. It's not as efficient, but I can do it a lot more quickly, and the ATP is ready to use immediately. So this provides a more explosive amount of force. The downside is that it produces lactic acid in the process. So it'll be necessary to remove this lactic acid in the circulatory system. That happens pretty quickly. So I've got two basic types of muscle cells, the fast twitch fibers and the slow twitch fibers. The fast twitch fibers are specialized for anaerobic respiration, whereas the slow twitch fibers are specialized for aerobic respiration. It's important to note that both cell types can do both types of respiration. It's just a matter of how good they are at one or the other when we ask them to do so. So the fast twitch fiber types contract very quickly and produce large amounts of force, but they also fatigue quickly. The cells look different from the slow twitch ones. They're larger in diameter, partly because they have larger reserves of glycogen, but few mitochondria. The slow twitch fibers are slower to fatigue. They can't generate as much force, but they can do so for a longer period of time. They are smaller, and they've got more mitochondria, and we can also identify them by their high oxygen supply. That includes lots of capillaries near the cells, as well as a large amount of this protein myoglobin. This is an oxygen-binding molecule that helps muscle cells to pull oxygen away from hemoglobin and into the muscle cell where it's needed. Before I move on, you may have run into this protein before. Cardiac muscle cells also produce myoglobin. And if somebody's had a heart attack, when those cardiac muscle cells get damaged, they tend to release their myoglobin into the bloodstream. This protein should only be found in muscle cells. So if you detect it in the bloodstream, that's a sign that muscle cells have been damaged. And if it's cardiac myoglobin, that's a sure sign that a patient has had a heart attack recently. I'm not going to say much about the intermediate fibers, other than they're a mixture of the two. So let's focus on the fast twitch and the slow twitch muscle cells. In a turkey, we find regions of muscle that appear more whitish and other regions that appear darker. This is the difference between light meat and dark meat. The pectoral muscles are white meat. These are mostly fast twitch muscle fibers. So the breast muscles are really large because these muscles help to get the turkey airborne. Of course, this turkey over here probably never got airborne, but a wild turkey or a chicken can get airborne very briefly. It takes a huge amount of energy, so we're going to rely on a large number of fast twitch muscle fibers to get this turkey airborne. But they'll fatigue very quickly, so it won't be able to fly south for the winter, maybe just up into a low tree branch. The legs, on the other hand, look different. The meat here is darker because it's mostly slow twitch fibers. There's a lot more blood supply to the muscle cells here. It doesn't take nearly as much force to keep a turkey walking around, so we don't need so many fast twitch fibers. We want to keep walking around all day long, so there's a lot of slow twitch fibers. These two tissues will taste different based on the amount of glycogen. Large amounts here in the fast twitch area, much less here in the slow twitch area. Human muscles aren't like turkeys at all. Most of our muscles are roughly 50% fast twitch and 50% slow twitch. If we look at some exceptional people, though, we can see differences. Long-distance runners tend to have more slow twitch fibers in their muscles, whereas weightlifters and sprinters tend to have more fast twitch fibers. So that raises the question, is this due to nature or nurture? On the nature side, I think some people definitely inherit genes that are going to make them more likely to be good at wrestling or weightlifting distance. But then with training, we can convert fast twitch fibers into slow twitch, or slow twitch into fast twitch. For instance, you may be a pretty good long distance runner, but as you keep training aerobically, some of your fast twitch fibers may be converted into slow twitch, and you get even better at that type of activity. There's one place where we do have muscles that are light meat and dark meat, and that's in our calves. The big fat gastrocnemius is primarily fast twitch fibers. That's good for jumping, whereas the skinnier soleus is mostly slow twitch fibers, and we rely on these muscles more for walking. Otherwise, the rest of our muscles are probably somewhere in this region here. Now, anatomy textbooks aren't terribly clear on how effective converting fast twitch into slow twitch or slow twitch into fast twitch muscle fibers is. I was once at a party, though, and got to talk to somebody with a PhD in physiology who worked for Nike. It was his job to design the workout regimens for some of the highest paid athletes in sports. I figure if there's anybody who's an expert on the subject, it was this person. And so we geeked out a little bit over physiology. And I asked him, and he said, well, we can't really be sure, blah, blah, blah. But then I said, well, what's your thought? And he was pretty certain that training certain ways could change the muscles and convert one type of fiber into another type. In fact, sprinters may already understand this. A number of sprinters will go through a tapering off period before a big meet. That would be enough time for muscle cells to convert from slow twitch into fast twitch due to inactivity without actually getting any weaker. So this might make you a faster sprinter on the day of the meet. It's hard to know for sure, but it's certainly an interesting idea. These pictures here just represent the fact that these two types of muscle cells are indeed different. To identify these cell types biochemically, or you can just look at their size. There's some very large cells over here, and then smaller muscle cells. We're looking at these cells coming out towards us, so we're checking out the diameter. So those were the sources of ATP generated by aerobic or anaerobic respiration. And we talked about the difference between slow twitch muscle cells and the fast twitch muscle cells. This would be another good spot to take a break, grab a drink, come back in a few minutes. Okay, welcome back. We're continuing on the topic of muscle strength. One question that we have is how can muscles generate a small amount of force to lift small amounts of weight versus generate a lot more force to lift something heavy? We need to be able to transition between the amount of forces that these muscles generate and do so smoothly. To understand this, I need to take a step back and discuss what the word twitch means. We already used it to discuss fast twitch and slow twitch muscle cells. Now in medicine, a twitch is usually something that happens to our muscles when they start to spasm. That's not the type of twitch we're discussing now. In science, when we say twitch, we're talking about something very basic. Some of the oldest experiments in muscle physiology were done by a man named Galvani. He took muscles from frog legs, dissected them while the muscle tissue was still alive, but separated now from the frog, and he measured how much force that these muscles could generate when he applied small electric shocks to them. So he was studying the animal electricity. Hopefully this sounds a little bit familiar to you. If not, you should go back and read Mary Shelley's Frankenstein, because it's based off of Galvani's work. Anyhow, what Galvani noticed is that sometimes when the frog muscles were just sitting there, not getting stimulated by any electricity, they would generate tiny twitches. And he noticed that the smallest twitch was a very repetitive amount of strength. It turns out these little twitches were caused by a single vesicle of acetylcholine being spontaneously released by the nerve fiber that was still attached, causing some muscle cells to contract. Since vesicles are all roughly the same size, all of these tiny little twitches were all the same size. That's where we get the term twitch when it comes to fast twitch and slow twitch muscle fibers. A muscle cell is capable of generating different amounts of force. But at any given time, a muscle cell is either undergoing contraction or not. The amount of force that that muscle cell can produce depends on a number of different factors. One of the first ones is called length-tension relationship. And it turns out that muscles, when they're partially flexed, are stronger than when they are outstretched. And that's because when muscles are partially flexed, there's an optimal amount of overlap between the actin and myosin. When muscles are stretched to their fullest, there's very little overlap between actin and myosin. And for that reason, partially contracted muscles are actually stronger than when they are fully outstretched. If you've ever been to the gym and had a spotter help you with that last biceps curl by just giving you a little bit of nudge at the start of your motion, but you were able to finish the rest of that curl on your own, you have experienced length-tension relationship. Your muscles weren't very strong at the beginning, but they got stronger the more bent that they were. Next up, we can regulate how much force a muscle produces based off of how many times we tell it to contract. Every time we release a little bit of acetylcholine onto the muscle cell, it will undergo a very small contraction, a single twitch. But the acetylcholine is removed very quickly, and if we don't keep releasing it, the muscle relaxes. But if we tell it to keep twitching over and over and over again, that muscle cell will generate more force. We might tell this muscle cell to contract a couple hundred times in a second. The reason that increasing the frequency of contraction increases the strength of contraction is that these twitches sum with one another. A single twitch would involve the muscle contracting and then relaxing. But if we increase the frequency of stimulation, we'll see that before the muscle has a chance to relax, it'll be asked to contract once again. As we increase the frequency, all of these contractions should add on top of each other, and the amount of strength that this muscle produces increases. The brain can send higher frequency signals or lower frequency signals to this muscle to generate different amounts of force based on this principle. Next up is the TREP effect. It turns out if we ask a muscle to contract and do it again and do it again, it actually gets a little bit more efficient with subsequent contractions. This is called the TREP effect, and it has to do with calcium. It turns out that when you ask your muscles to do, let's say, that first push-up, it's kind of hard. But they do it, some calcium is released, and that causes a contraction. And as we keep on doing a few more push-ups, they get a little bit easier. And that's because we keep releasing the same amount of calcium, but it's not being returned to the sarcoplasmic reticulum at the same rate. So calcium is building up in the cytoplasm with subsequent contractions. More calcium means more troponin and tropomyosin are moving out of the way, which means more myosin is binding to actin. And when that happens, we get a stronger contraction. So the TREP effect is a stair-step increase in muscle contractions as we continue to do the same activity. Of course, this doesn't last forever. I can't just get better and better as I do more and more push-ups, and that's because fatigue will eventually set in. This is how I remember the TREP effect. If you don't know who the Von Trapps are, this won't help you. But this illustrates a good way of remembering boring facts in anatomy class. Come up with a rhyme or something silly, a goofy picture. Your brain will do better at remembering something funny than it will something boring. And if you can just give your brain a little bit of a hint, it'll be better at pulling out the boring information. Let's review. A single muscle cell can generate different amounts of force based off of how stretched it is, based off of the frequency of signals that are coming from the brain. That will lead to summation. Lastly, the TREP effect may also be coming into play. Next up, we're going to talk about how an entire muscle can generate different amounts of force. And to do that, we'll talk about motor units and recruitment. Those are two pretty important topics. Let's start with motor units. A motor unit is a single neuron plus all of the muscle cells that it controls. When this neuron fires an action potential, all of these muscle cells will contract. So this is a group working together. It turns out that within a motor unit, all of the cells should be the same type of muscle cell, meaning they're all fast twitch or they're all slow twitch. Recruitment is our next topic. When a muscle is relaxed, It turns out that some of the motor units are actually firing. There's a small amount of tone, meaning that muscle is actually generating a small amount of force, even when we think of it as relaxed. But then when we ask that muscle to do something, like lift an object, it will undergo recruitment. My brain doesn't have to know exactly how much an object weighs. It just asks the muscle to lift it, and the muscle will increase the amount of force until it's enough. And when my eyes and other parts of my nervous system tell me, hey, that object's moving, I will stop recruiting and generate just that amount of force that's needed. So recruitment means that I'll fire a few more motor units. And if that's not enough to move the object, I'll fire a few more motor units and a few more until finally I'm generating enough force to move the object. This is not something that the brain has to think about. This is just what our muscles do when we ask them to do something like lift a weight or start running. I'll keep recruiting more and more motor units until the thing that I want to do is being done. It turns out that at maximal tension, the maximal amount of force that your muscle can generate, you're not using all of your motor units. So when my muscle is generating its maximum amount of force, it's using just some of its motor units. And that means that when one of those motor units starts to fatigue, it can begin to relax, and another motor unit will take over. And as long as this relaxation and taking over by the next motor unit happens pretty quickly, I'll have sustained tension in that muscle. As muscles fatigue, though, motor units can begin to wear out a little bit too soon, and it may take a fraction of a second for the next one to take over. And when that happens, we start to notice a wobble in that muscle, or a shake. One of the first things to change with exercise is a change to muscle tone, the number of motor units that are firing when a muscle is at rest. Somebody who has very good muscle tone, their muscles will be more flexed when they're resting than somebody with really bad muscle tone. This represents a quick change to the nervous system, not an increase in muscle strength. This slide here shows that at peak muscular activity, we're only using about half of our motor units. If I could use all of my motor units, I might lift a really heavy object, but then if all of the motor units fatigue all at once, I would then drop that object on my head, possibly causing injury. Similarly, if I used all of my motor units all at once, they might be able to generate enough force to tear my tendons. So the brain has a protective mechanism in that it's only capable of acting about half of the motor units, even when it's trying to exert the maximum amount of force possible. Now, many of you have probably heard stories of people undergoing feats of superhuman strength in an emergency. For instance, mothers needing to lift a car when their child is in danger, or something of that nature. It seems possible that humans could generate superhuman amounts of force in a dire emergency. That's not something that we can study in a scientific laboratory, so we can only rely on stories. Muscle tone is very important. It helps us to keep our body sitting or standing upright. This child over here is suffering from a very debilitating symptom called hypotonia. This is very dangerous. Muscle tone is the first thing that we lose with inactivity, and also the first thing that we gain with exercise. I'm totally certain that this dude's head over here was not photoshopped onto two different bodies, but we often see before and after pictures promising us that if you buy the right supplement, that within just a few weeks you can gain massive amounts of muscle tissue. That just doesn't happen. What can happen are changes in muscle tone. My ex-sister-in-law and her husband were both professional bodybuilders and firefighters. They were both in very good shape. They both got in car accidents right around the same time and both wound up in the hospital. Both of them got approached by makers of different muscle-building supplements and were asked to become the next spokesmodel for those supplements. They had to promise to not reveal their secrets, which they did to me, and now I'm revealing to you. Both my ex-sister-in-law and her husband worked out a lot. Let me tell you, they were both buff. That took years and years of exercise and hard work. But when they had to stop exercising because of their car accidents, they both lost a lot of muscle tone. And it was at that point that the supplement makers took a before picture. And then 4 weeks later, once they were given the okay to go hit the gym once again, they used some sort of supplement, hit the gym, and gained all of their strength back really quickly. And their after pictures made them look much, much more ripped. They didn't really have enough time to lose a lot of muscle mass, and they weren't gaining muscle mass back in those 4 weeks. What they gained quickly was muscle tone. The muscle mass was already there, it was just getting masked by their loss in tone. So, I would not expect the same results if I used the same supplement and hit the gym for the same amount of time. I just don't have that same amount of muscle mass that they did to see their results. So that's one of the secrets with these before and after pictures. We're usually seeing a change in muscle tone. People would not expect to see an increase in bulk. So muscles can generate different amounts of force. My brain can send a higher frequency of signals. We can also recruit more muscle fibers. It turns out I recruit the smallest ones first and then largest ones last. And this should hopefully generate the amount of force that I need. Back to our numbers for fun. How many motor units are there in a muscle? Well, out of the million muscle cells in the gastrocnemius, there's only about 500 neurons. So divide 1,000,000 by 500, and you've got the average number of muscle cells that each of those neurons controls. Nevertheless, that means that the gastrocnemius can generate 500 different levels of force. There's either 1 all the way up to 500 of those motor units. For the fingers, you might want even finer control over the amount of force that you can generate. And those smaller muscles may have even more motor units for a lot finer control. So those are some of the differences between generating small amounts of force versus larger amounts of force. We talked about the Trepp effect, length-tension relationship, frequency of stimulation, and then this last bit, recruiting more and more motor units. That's going to lead us to the last part of our lecture. How do muscles change with exercise? One of the first things we want to discuss are different types of muscular contractions. They can either be isotonic or isometric. Isotonic means the same amount of tone. Iso is the same, and tone means roughly strength. Isometric contractions means that the muscle doesn't change length. Its measurement, or its metric, stays the same. So an isometric contraction is where a muscle is generating force, but nothing moves. Whereas isotonic contractions are ones where we're generating force and something moves. Either the muscle gets shorter or the muscle gets longer. So isotonic contractions come in 2 basic flavors. The simplest one is if you generate more force than the weight you're trying to lift, the muscle should get shorter and the contraction occurs just like you want. This would be called a concentric contraction. If the muscle changes in length in any other way, we call that an eccentric contraction. For instance, if I do a biceps curl and I lift the weight, that would have been a concentric contraction. But if I try and resist the force of that weight, but let it down slowly, my muscle would be doing an eccentric contraction. There are other types of eccentric contractions. Let's say I try and lift something that's way too heavy, and my muscles are shaking from side to side. That's a different type of eccentric contraction. Isometric means there's no movement at all. I'm using my muscle, I'm burning up ATP, but nothing is changing in length and no motion is happening. Eccentric contractions tend to do the most damage to our muscles. This gentleman here, after running uphill, is now going to run downhill and is going to be very likely to give himself some shin splints as he does a bunch of eccentric contractions. Even worse would be trying to lift too much weight and having muscles move from side to side. Eccentric contractions can do damage to our connective tissues or to the muscle cells themselves, and when this happens, this can trigger inflammation, and that inflammation can lead to prolonged muscle soreness. So if your muscles are sore the next day after a big workout, or 2 days later, or as we get older, 3 days later, that's due to inflammation. It is definitely not lactic acid that's still there in your muscles. Lactic acid is removed very quickly. When lactic acid builds up in a muscle, that's the burn that you feel at the end of your workout, when your muscle is starting to fatigue and you can't do that last rep. That should go away fairly quickly. Prolonged muscle soreness, though, may last for days because inflammatory molecules don't go away. They recruit more white blood cells, which release more inflammatory molecules until the repair is done and the tissue is healed. One thing that compression garments can do is reduce eccentric contractions. Compression garments are spandex that's a little bit too tight. They do a number of things, but one of the things that they do is reduce injuries, and this has been proven pretty well. They do this by holding the muscle cells parallel, promoting concentric contractions, and reducing eccentric contractions. If I didn't have this, it might be possible for these muscle cells to move at angles from one another, which would tear the endomysium between them as they move in different directions, and this tearing would lead to inflammation. It turns out we need to do a little bit of damage to our muscles if we expect them to get stronger. Micro tears to our muscle tissues will trigger repair and growth. Too many micro tears might lead to an injured muscle, and you might have to take it easy on that muscle for a number of days, in which time the muscle could actually get weaker. This was something that Bill Bowerman, the head of the track department at the University of Oregon, happened upon. I don't think he understood the biology, but he understood the end result. At that time, if you wanted to have a better track team, you just told your athletes to work out harder, come in on weekends, work out every day. What Bill Bowerman did is gave his athletes a day off. They worked really hard one day and then had a day of rest. And during that time, after having damaged their muscle cells on the workout day, gave their muscles time to not only repair that damage, but to actually increase in strength. And for years after developing this type of workout routine, Oregon was known as Track Town and dominated in the track and field area. We now understand exactly what's happening. We need to give muscles time to not only repair the damage that you've done from a workout, but to get stronger as well. So that's going to be our next topic. How do muscle cells get stronger? And that's going to depend on what type of activity you have asked them to do. Aerobic activities will lead to different changes in muscle tissue than anaerobic activities. We'll also be using our fast twitch and slow twitch fibers differently, depending on which type of activity we're doing. Aerobic activity, or endurance sports, rely heavily on our slow twitch muscle fibers. When we do a lot of this type of activity, we will tend to increase the number of mitochondria and increase the oxygen supply to these muscle cells. Whereas anaerobic activities rely more heavily on our fast twitch fibers. And when we do these types of activity, the muscles will actually increase in size. That's why bodybuilders typically do heavy activities, lifting heavy weights rather than doing endurance training. If larger muscles is the goal, you better do more anaerobic type of activities. So, with aerobic activity, muscle cells will tend to increase their blood supply. They do this by activating angiogenesis, which causes new capillaries to grow into the area. They can also increase their number of mitochondria, and they can increase their amount of myoglobin, which makes the cells even darker. Anaerobic activities can lead to an increase in the number of myofibrils, and with an increase in the amount of actin and myosin, that leads to increased strength. These cells will also increase their glycogen reserves, and for these two reasons, the muscle cells get larger in diameter. Both fast twitch and slow twitch muscle cells can undergo both aerobic and anaerobic respiration, but they don't do so as well as one another. Let's say I've got a muscle with just two cells in it, a fast twitch and a slow twitch muscle cell. And if I ask this muscle to do aerobic activity, both of these cells can do so, but the fast twitch cell is going to get tired sooner. So at the end of my workout, the slow twitch fiber would have done a lot more work. And for that reason, the slow twitch cell is going to undergo more changes than the fast twitch cell. It'll increase its myoglobin content, increase its mitochondria, and increase its blood supply. Whereas the fast twitch cell, it didn't do so much work, it's not going to see big changes. On the other hand, what if I asked the same muscle to sprint or to lift a really heavy object, to do an anaerobic activity? Both muscle cells can be pushed to peak performance. They can both generate the maximum amount of force possible. It just so happens that my fast twitch muscle cell will be generating a lot more force when I ask it to generate the max than will my slow twitch muscle cell. Therefore, the fast twitch muscle cell had a better workout. So it's going to be the one that responds more to this type of activity. And it does so by making more actin and myosin, storing more glycogen, and increasing in size. So muscle hypertrophy can happen by a few different mechanisms. We're always going to lead to more actin and myosin in the muscle cell, but there's different ways this can happen. One way is simply transcribing the genes for actin and myosin from the nucleus and translating them out in the cytoplasm. That way you could get more actin and myosin, and that muscle cell would now be larger. Sometimes, though, we require the help of stem cells. One of these myoblasts may migrate into the area and join up with this original muscle cell, making it larger. Both ways lead to a stronger muscle cell. It turns out that both of these processes are given different names. I'm less interested in those names than I am what happens. With hypertrophy, we get cells growing larger due to DNA transcription, whereas hyperplasia involved something similar to cell division, where myoblasts came in and joined up with an existing muscle cell to make it larger. Either way requires two signals. I needed to have damage to the muscle cells in the form of small amounts of microtears triggering some inflammation. But I also needed hormones. If all I needed was microtears and inflammation, I would get stronger every time I got an infection, or I could get stronger simply by slapping myself. That can damage muscle cells. But if that's all that happens, I just repair that damage, I don't get stronger. With a workout, I not only damage my muscle cells, but I also produce hormones like testosterone and growth hormone. And when you get both of these things, the hormones plus inflammatory molecules, you not only repair the damage that happened, but you try and make the muscle stronger so that the next time you ask it to do the exact same thing, it doesn't get as damaged. Hopefully that sounds similar to what happened in bones. When we repaired a bone fracture, we didn't just repair it to the way that it was originally, we actually made it a little bit thicker and stronger in that spot. Same with skin. If I leave behind scar tissue, that makes that skin a little bit tougher in that spot. It's not as stretchable, but it is stronger. So muscles that have undergone a significant amount of exercise can generate the hormones testosterone and growth hormone, and this can lead to an increase in muscle strength. These hormones can also be injected directly into the bloodstream. This would be an example of somebody abusing a performance-enhancing drug, but this can also lead to hypertrophy, especially if matched with a workout routine. We know that levels of these hormones rise in the bloodstream naturally after a significant amount of a workout. Based on this, people came up with a theory of multi-joint exercises. That if you want the biceps to get really big, it might not be enough to just work out the biceps. The theory is that if you also worked out the quadriceps would produce a lot more growth hormone and testosterone than the biceps alone. And that maybe some of the hormones coming from the quadriceps would help the biceps to get even stronger. It turns out the experiment's been done, and at least at this point, based off of one small experiment, evidence suggests that working out the legs is really only good for the legs. If you ask two groups of men to undergo exercise where they work out their biceps, doing the same amount of biceps curls, but one group also does a leg press exercise, the ones who did the leg press exercise will have much higher levels of growth hormone and testosterone in their bloodstream after their workout, but it does not lead to an increase in strength of the biceps. So at least at this point, the multi-joint exercise theory, while it was sound, doesn't seem to work. That's not to say that if you work out your upper body one day and then work out your lower body the next day, that that's not a good idea. That's in fact a very good idea. You're giving one part of your body a rest while working out the other part. Just don't expect that to be really good for the biceps, any better than just working out the biceps alone. So let's summarize. If I ask a muscle to do some exercise, if it does anaerobic activity, it's going to be mostly the fast twitch fibers that are doing all of the work. Therefore, they're going to be the ones that generate the most hormones and get the most damage. So in response to a little bit of inflammation and local production of growth hormone and testosterone, it's going to be the fast twitch fibers that respond the most. They will make more actin and myosin, and they will have more glycogen reserves, which makes them larger. On the other hand, if I ask the same muscle to do an endurance activity, it's going to be the slow twitch fibers that do most of the work. And therefore, they're going to be the ones that respond the most. We'll still get inflammation, and we'll still get local release of hormones, but the muscle cells that did most of the work are the ones that know to undergo the biggest changes. They will increase their amount of myoglobin and mitochondria, and will activate angiogenic signals. And the last thing there that you might have noticed is we might convert some of the fast twitch fibers into slow twitch fibers. I would be better at endurance now, although I wouldn't be quite as strong. Now, before I move on, I just want to point out that you don't literally have to injure your muscle cells every time just to get them to increase in strength. Depends on how much of a strength increase you want. For your elderly patients, for instance, You're not going to be pushing them to the brink of exhaustion. But a little bit of activity for them can help to increase their muscle strength, even if they don't have aching, sore muscles the next day. A little bit of inflammation may not be so painful, but it can lead to a little bit of increased muscle strength, and that's often very beneficial for your patients. On the other hand, if we don't use our muscles, they will get weaker. The first thing that they lose is tone, but over enough time, they can start removing actin and myosin and losing muscle strength. It really takes weeks to months to start losing muscle strength. That won't be happening terribly quickly. But that's something that you might see in cases where people's muscles become paralyzed, either partially or fully, such as with polio, ALS, or some other diseases. For instance, NASA understands fairly well that if you launch astronauts into outer space, they're going to lose about 20% of their muscle strength in just 2 weeks. For somebody who is bedridden, you might see the same thing. Or if somebody's in a really hefty cast, you're going to expect the same loss of muscle tissue over time. Now, for astronauts, they can do resistance training in space. That means using things like rubber bands, which don't rely on gravity to provide weight. It's still not perfect. This decreases the loss of muscle strength, but doesn't stop it entirely. On the space station, astronauts were still losing a percentage of their muscle mass over months. Next up, what about stretching? Stretching is very healthy for muscle tissue and tendons. It tends to increase the length of these organs, which increases range of motion, which decreases risk of straining the tendons or muscles. It just so happens it doesn't matter when you stretch. You could do it before a workout or after. It's healthy either way. Now, one thing that's a big deal in physical therapy and occupational therapy is this process called proprioceptive neuromuscular facilitation. That's not a name I expect you to remember, but these other two things are things we've already covered. Passive stretching would mean stretching a muscle, not on your own, but having somebody pull on you. And then while that's happening, you will flex your muscles to generate an isometric contraction. That just means you're flexing your muscles using ATP without actually moving your limbs. And it turns out the combination of these two things is much better than just stretching out on your own. So while this is a big deal in physical therapy and occupational therapy to help decrease the risk of injury in patients, this is also becoming popular in sports and sports medicine, for it's much more effective than just stretching on your own. Next up, another thing that can cause muscle cramps is loss of electrolytes. It turns out those electrical signals that I talked about earlier involved sodium ions rushing into the muscle cell and potassium ions rushing out. When that happens, the muscle will contract. To get us back to a relaxed state, we need to get rid of this electrical signal, and then calcium will do its thing. So sodium will be pumped back out and potassium back in. If I keep on working out this muscle, more sodium will rush back in and potassium rushes out. But if I keep on working out really hard, I may sweat away a lot of those potassium ions. And when that happens, I can't get the sodium back out, and this cell remains in a state of contraction, or a cramp. Replenishing my potassium levels can help, like eating a banana or drinking Brondo, which has electrolytes. A little bit of massage might help too. My entire body probably isn't out of potassium, just this localized area. So if I push fluid around, I may help bring in some more potassium to this area. The last topic is what else happens when we exercise muscles. Turns out I can not only do damage to my muscle cells and connective tissue, which might release inflammatory molecules when that happens, they also release reactive oxygen species, or oxidative stress. So these reactive oxygen species are free radicals, which can be nasty. Free radicals can damage DNA, which could lead to mutations which lead to cancer. And free radicals can also damage lipids, which makes them sticky and more likely to stick to my arteries, causing heart disease. So I don't like having free radicals in the wrong place at the wrong time. On the other hand, I absolutely like having them in certain places. For instance, when we are using oxygen to break down glucose, I need to have free radical intermediates. Free radicals can also be generated by UV damage. That's not really good. But they are also released by white blood cells to damage and kill viruses and bacteria, or to even kill off cancer cells. And my saliva and tears are full of reactive oxygen and reactive nitrogen species, which also helps to keep pathogens in check. So as you can see, there's some places where reactive oxygen species are really good, like my mitochondria, and some places where they're bad. So an antioxidant is something that can neutralize reactive oxygen species. So are these reactive oxygen species always bad? Well, yes, sometimes they are. There are a few diseases where oxidative stress can cause even more damage, like diabetes and Alzheimer's disease. And in these cases, taking an antioxidant supplement has been shown to be beneficial. But these are not your average human beings. For a healthy person, oxidants are actually really good things. Heck, I can't live without oxygen. I wouldn't want antioxidants getting rid of all of my oxygen. It's true that oxygen can damage tissues, but that's what I've got hemoglobin and myoglobin for. Oxygen doesn't just float around my body going wherever it wants. It only goes where I tell it to. And reactive oxygen species can be produced when I damage muscle cells. If I exercise a little bit too much, some of those mitochondria might burst open, releasing some reactive oxygen species that should have been contained within the mitochondria. Turns out, these reactive oxygen species can trigger angiogenesis, and that can decrease my risk of heart disease, because if I have more blood vessels around my heart will be healthier. So it turns out that antioxidants would actually not be healthy for a healthy person to take, and there's very clear evidence for this now. Taking antioxidant supplements actually increases the chances of somebody dying early. And it turns out those antioxidant supplements block the ability of muscles to get stronger. We actually need those reactive oxygen species to be released for a muscle cell to know that it needs to repair that damage and grow stronger afterwards. And antioxidant supplements have been shown to reduce the ability of muscle cells to grow stronger following exercise. And as you should know, exercise is very beneficial. It not only makes our skeletal muscles stronger, it makes our cardiac muscle stronger. And by increasing angiogenesis, I've got more blood vessels. That means I'm at a much lower risk for a heart attack. Now, many fruits and vegetables are rich in molecules that are antioxidants. Everybody should be eating more fruits and vegetables. What this research over here looked at was supplements, which contain the same amount of chemicals in the equivalent of, say, thousands of oranges. We could never physically fit 1,000 oranges into our stomach all at once, so we will never get the level of antioxidants from eating healthily that could cause these adverse effects that we see with supplements. So feel safe in telling your patients to eat their fruits and veg. That's absolutely good for us for any number of reasons. But they could probably stay away from the pills. So far, evidence for healthy people at least suggests that we really don't need to be taking these pills. So to summarize, with certain diseases, like diabetes and Alzheimer's disease, this can lead to damage to our cells, which release reactive oxygen species, which can damage blood vessels, which can increase our risk of dying from cardiovascular disease. But with exercise, damage to our muscle cells releases some of these same reactive oxygen species. But the environment here is very different. It turns out when this happens, it actually leads to a reduction in atherosclerosis and an increase in muscle strength. And when that's your cardiac muscle, that will lead to a lower blood pressure overall. And that actually reduces the risk of cardiovascular disease. So this may seem complicated, and it is. The human body is complicated. If anybody ever tells you it's simple, they're lying or they're just too dumb to understand the reality of the human body. Here, what I'm trying to get at is that a little bit of damage to our muscles is actually beneficial. And part of that involves the release of reactive oxygen species from our mitochondria. So, just because something can be bad for the body doesn't mean that it's always bad for the body. And blocking reactive oxygen species broadly with a high dose of antioxidants from a supplement actually does more harm than good. So that wraps up the lecture on muscle tissue. Be sure to go back over the difference between slow twitch and fast twitch muscle cells and how these two different cell types respond to exercise differently and why that might be healthy, either way, for the human body. We also talked about motor units and muscle tone and different types of muscle contractions. But that should wrap us up for the day.